But I'm Matthew Lera. I am the exploration project scientist for biological and physical sciences, talking about tissue chips on Artemis 2. Kevin Sato is the exploration program scientist, and he's sort of, I'm his deputy. He was going to talk today, but was out on leave, so I'm jumping in, and you're stuck with me for a couple slides. So next slide, please. So this is just sort of an overview of tissue chips and high level of what we're hoping to accomplish as we go deeper into space, using tissue chips as a surrogate for human biology. The objective is to use these 3D tissue chips to obtain data, looking particularly at the combination of two stress factors, reduced gravity or altered gravity and space radiation. So the tissue chips gives us an opportunity to test those two variables together, actually in environments that the crew will experience. So we're the sort of overarching further out goal of utilizing these tissue chips is to study them in the lunar environment, using tissue chips that are actually made from human cells. So you can extract human cells and put them into the tissue chips in culture. So you're actually probing cells that have been extracted from humans as a surrogate representation of the human themselves. And then eventually we want to send these tissue chips further out beyond, Artemis 2, and eventually all the way to Mars, looking particularly at the transit environment, and then eventually down to the surface of Mars. So Artemis 2 is the first step in building that data set, building out the knowledge set that we need to enable the deeper space human exploration. And so again, the data that we developed from Artemis 2 will help us study spaceflight stressors at more of a mechanistic level that can't be accomplished by studying directly on the human. And they'll help us investigate mitigation strategies to support sustainable presence on the lunar surface and eventually onto Mars. And then it'll also allow us to eventually get into personalized medical kits. So these, if we have cells that we can probe that are specific to individual crew members, we can actually use them to plan individual medicine mitigation for future missions that they would be on. And then of course there's always the tissue chips are sort of an emerging thing that are a supplement to whole organism research, and so there's a lot of human benefits down here on Earth that this will lead to as well. So next chart, please. A little bit more about the science here. Again, the lunar environment that we want to look at is the combined stressors of the altered gravity and radiation environment. Particularly looking at the mechanisms of acclimation and age-associated diseases, and then look at evaluating some countermeasures on future missions. So expected outcomes here, we want to understand the impact of spaceflight on biology and the ability of humans to survive in these environments, so that we can plan and enable using current human exploration missions to enable future human exploration missions. I think I talked about those last two bullets. So next chart, please. So the tissue chips that we're going to be using for Artemis 2 are these Emulate Bio tissue chips. This is a company that puts out commercial products. So these tissue chips are in the middle of our payload. They're composed of a clear, flexible polymer. They're about the size of a USB member stick, maybe a little bit shorter than a microscope slide and thicker. And they have these hollow microfluidic channels in there. So within those microfluidic channels, we'll have bone marrow cells that are extracted from humans for Artemis 2, and then the chips themselves are hooked up to a fluidic system, and then inside our payload, they're in a CO2 controlled environment, constantly getting media flowed to the cells to maintain them throughout the mission and incubate it to the right temperature. So we're using these to, again, analyze the biological and biophysical mechanisms that underlie physiology and disease development, and these will enable investigations of mechanisms. These enable investigations of mechanisms at the cell tissue and organ levels. The next chart, please. So this is specific to Artemis 2 hardware and concept of operations here. So those tissue chips are in the middle of this payload. The payload that we're flying for Artemis 2 is a version that has flown on ISS before. So it's been optimized by Space Tango, puts out their Cube Lab, and they've flown tissue chip payloads on ISS before. So they're developing a new version of this, of their Cube Labs specifically for Artemis 2, since the configuration is a little bit different. It'll be completely autonomous and have internal battery power. So we're not getting power from the Orion, we're not getting comms from Orion. Will be completely self-sustaining, and there'll be 4 of those emulate tissue chips in the middle of this for the duration of Artemis 2. So the payload will fly alongside the crew inside the Orion capsule and experience the same altered gravity and radiation environment that the crew sees. And then, so upon splashdown, the hardware will be removed and put into the ship for a return back to the port, and they'll actually we'll get some power from the ship, and at that point the payload will know to administer a fixative so that the cells will be fixed shortly after splashdown, and then they'll continue the ride back to the port. We'll do some sample processing once we get to the port and ship those samples back to the science team in their lab, and they'll do some single cell RNA sequencing on the cells once they get back to their lab. So that'll allow us to look at gene expression changes that are associated with the mission overall. So this picture here is we were at JSC last week, actually. We were doing some integration and safety testing there, and we got to get into the Orion mockup and do a fit check. So you can see this weird shape is for a reason. It fits in that specific location, and it's no longer a cube; it's more of a trapezoid shape. So we got to do a fit check with it, make sure that it's securely fastened into the spot that it's going to be for Artemis 2. And then the operations, the crew, it's pretty minimal. There's a LED series of LEDs on top and a button that gives a status and just some housekeeping data that the crew will just hit that button and then call down and report what LEDs are lit and what's not lit, and then our team can translate that into how the payload is doing in real time. So it's pretty minimal crew time, pretty minimal resources that we need to do this. But it's a pretty there's a lot of science going on inside that box, and we'll get great data out of this, and hopefully enable some future versions of this on future Artemis flights too. The next, that's my last chart. I think there's just a thank you. So yeah, that's it for me. I guess I finished a little bit early, but again, this is great science that we're hoping to accomplish with this, and proving out that the tissue chips are a great tool for this kind of research that we can do deeper in space.